Okay, students, right? Very good morning. I hope all of you can hear me, right? So today we are going to start learning genetics, right? A new unit, unit number 6, and one of the most important unit in your syllabus, right? So genetics, the most important thing is, Pute, that there are some basic principles, right? Basic principles, and it is somewhat related to mathematics also. So biology, the science of the bioorganisms is correlated with some calculations, right? So that is genetics, and this is very easy. The most important thing is to understand the basic principles of genetics, right? So, uh, right, hope all of you can hear me clearly, right? Uh, if anyone is not clear, please put a message in the chat box. Right. Okay. Right. Yes, sir. Right. Thank you. So, uh, right. Genetics. So this is unit number 6, and we'll go one by one. Very slowly, we'll try to understand the basic principles, the theories, and the scenarios behind this topic of genetics. Right. Now, to start with, what is genetics means. Now, for the existence of the organisms, one generation create another generation, right? So that is how the existence of the species goes on, right? Now, some of the features that one generation is having, right? We'll take generation 1. So some of now the features of that generation is transmitted to the next generation, to the generation 2, right? Right. So you take the population, right? Now, what do you mean by population? Can you remember the basic principles that we learned in unit number 1? The hierarchy of bioorganisms, starting from the biomolecules, then the cell organelles, cells, tissues, organs, organ systems, organisms, and then population, right? So population means, Pute, a group of organisms which in the same species which lives in the same environment. Now you take the candy, right? You have the candy population means the same species. That means Homo sapiens sapiens living in candy area, right? Now you take the human beings living in the candy area, right? Are all these humans same in their outer appearance? No, everyone is different to the other people, right? So you call it variation. There are some variety of organisms. You learn about the variety, right? The variety. There is a variety among the organisms in the same population. There are some differences, right? Now, some of these variations are inherited to the next generation. For example, right, my skin color, my contexture, my height, and my appearance, some of my hair color, my color of my eyes, the nail patterns, all these things, they are inherited to the next generation. There are some variations which are not inherited to the next generation, right? Now, for example, sometimes they say very good parents are having very naughty children. Sometimes very bad parents are having very good quality children. So those are not inherited to the next generation, right? Now, in genetics, focus only on the features which are inherited from one generation to the other generation, right? So these variations. Now we divided the variations into two categories: inheritable variations and non-inherited variations, right? So inheritable inheritable variations are transmitted from one generation to the next generation, right? Now, how it happens, how these inherited variations are transmitted from one generation to the next generation. That mechanism, right? That theory is called as genetics. Simple as that. Okay. So what is genetics? Now take into your mind, understand what is genetics. Genetics means in a population, you have various type of variations. Some of these variations are inherited to the next generation. That means that those variations are uh, goes on with each and every generations. So how these inheritable variations are transmitted throughout the generations. That mechanism, how it happens, is genetics. Okay, right. Now, the mechanism of these inheritable variations, how it goes from generation to the generation, is first invented or found by a Austrian Augustinian monk, and his ethnicity Augustinian, mean monk, right? Who is that great scientist? He is the Gregor Mendel. Gregor Mendel, right? He's a Catholic monk. He is the one who did various type of experiments and found the basic principles, right? Basic theory for the first time in the world, how the inheritable variations goes from generation to the generations, right? So therefore, Gregor Mendel is known as the father of modern genetics, right? Now, the important thing, by the time when he finds the theories behind the genetics, the chromosomes or the DNA, nucleotide, none of these are not found in the world, right? So before that, before the invention of the chromosome, before the invention of a gene, before the invention of the DNA pattern, he invented this thing, right? So he doesn't know about a name called gene. Therefore, he used the instead of a gene, he used a word called inheritable factor, right? By using that word, he explained very truth about the mechanism, how the inheritable variations transmit through the generations. Okay, understood. Right. So Gregor Mendel, remember the name, right? Now we'll see uh, the basic theories behind this, the basic mechanism behind this genetics story. And before that, today, there are some important words which are specific for the field of genetics. So before you learn the theory, before you learn the mechanism of genetics, you should know some of these vocabulary, right? Some of these scientific technical terms used in genetics, right? So first, we will learn what are those words and we learn the vocabulary, then it is very easy to understand the mechanism. Understood? Right. So I am teaching you from the very basics of the genetics. Okay, right. Now we'll see the vocabulary. What are the specific technical terms that we use in genetics? Okay. Right. This appearance is somewhat different because, before a few days back, 1 week back, I came to Malaysia and I am teaching you in Malaysia, right, in my apartment. Right. I had to find some of these learning materials and these things to set up. And so all these things, I hope that the the display is more clearer than what I did at Sri Lanka, right? So if you have any questions, same as the previous classes, right? Any questions, any clarification, you just put a message and I clarify it. And the students who were not able to watch the record, the class, not who were not able to join the class in live, right, can see the recording. I'll share it. Someone has put a message. It's great. All of you are participating. Huh? Very good. Uh, right. Okay. Right. Good. Can hear and good morning, sir. Good morning, Gagi, and good morning for everyone. Okay. Right. Okay. Right. So we learn some of the technical terms that are used in genetics, right? So we call it vocabulary. Vocabulary in genetics, right? So now I told you that in genetics, we are learning the variations which are inherited from one generation to the other generation. Give me 1 second, I have to plug on this charger. All right. Okay, right. So inheritable variation. We are focused only on the variations which are inherited from one generation to the other generation. Inheritable variations. Inheritable variations. Right? Okay. So now we'll take one characteristic. For example, we'll take the hair color of human beings. Hair color. There are some various hair colors among the human population. Some are having black hair, right? Some are having brown hair, right? So usually Sri Lankans are having brownish black hair, right? Some are having very dark black hair, some are having brown hair, and some having black hair is tinted with brown hair for the attraction, right? You know, the actresses and the cute girls, they like to have their hair in brown color, right? And also some are having blonde hair, the ash color hair, especially the European people, right? So these are the variations if you consider the characteristic of hair color, right? You take the eye color. To take the eye color, there are some people who are having brown eyes and blue eyes and green eyes, right? So again, the girls, when they are participating to celebrative events, they some uh, the lid like thing and change their color into the blue and the green, right? So forget about that thing. What I'm talking about the natural variations that are there in the population, right? So eye color, you have the brown, blue, and green people are there, right? Now, this feature, hair color, or the eye color, skin color, right? The height or the nature of the ear, whatever the thing, right? The characteristic or the feature that we are focused on is called as character in genetics, right? So hair color and the eye color here is known as character. So hair color is a character. Eye color is a character, right? So in that character, you have different variations. Brown hair, black hair, blonde hair. Now these are called as traits. traits. So under the character of hair color, black, brown, and blonde colors are the traits. So under the character of eye color, brown, blue, and green are traits. Understand? Right. So in vocabulary, we learn first of all two words: character and traits. Right? Character means the specific feature that we are focused on, which are inheritable. So hair color is a character, eye color is a character. Under a character, the variations, each and every individual variations are known as traits. So black is a trait, another trait is brown hair color, another trait is the blonde hair color. Eye color, brown eyes, one trait, blue eyes, one trait, green eyes, another trait. Understood clear. So in the vocabulary, we learned two words, character and the traits. Clear? Right? I hope all of you understood that thing. Right? Okay. So now in this scenario, the black hair, brown hair, and the blonde hair, we can see from our eyes. These traits are observable. You can clearly see a person with black hair, a person with brown hair, right? You can see a person with blue eyes, a person with green eyes. You can see. So these traits are observable. So the observable traits are known as phenotype. They are known as phenotype. So these are phenotypes. Understand? Right? So what do you mean by phenotype? The variation that you can see. So black hair is a phenotype that we can see. How that person who is having black hair, how he got black hair is written in his chromosomes. Right? Now, a person with black hair, can you see the chromosomes and the genes? When you look at the person, this person is having black hair, see that his DNA is there. Can you see the DNA cells or no? You cannot see the gene or the chromosome, but because of that gene, he got black hair. Understood? Right. Now, at the time of Gregor Mendel, the people were not aware of a gene or a chromosome or a DNA. Therefore, Gregor Mendel used another name instead of gene. Right? That name is inheritable factor. Inheritable factor. Right? So, these days in modern genetics, we don't use the inheritable factor. We are talking about a gene responsible for black hair. That's all. But at that time, Gregor Mendel didn't know about this word. So he used a name, inheritable factor. So inheritable factor is there inside the cells, which you cannot see. That is called as the genotype. So inheritable factor, or the thing that is there in the chromosomes or the gene, is called as the genotype. So we learn another two words. First two words we learn character and the traits. Clear? Right. So these traits are observable in one trait. There is an observable part, there is an invisible part. Right? So observable part is the phenotype, what you can see. So phenotype means what you can see outside. Observable. Right? So that is appeared because of his cells or the chromosomes are having specific sequence of DNA, right? That is genotype. So the fourth word is genotype. And Genotype is there because of the inheritable factor, right? So number 5 is inheritable factor. So inheritable factor, today we call this as gene, but at the era of Mendel, he used the word inheritable factor. Okay, understood, right? So from the very basic, now we learned 5 words in genetics. What is a character? What is a trait? What do you mean by phenotype, and what do you mean by inheritable factor, and what do you mean by the genotype? Okay, right. Right. Krish also has joined. Okay, good morning. To you, very good morning and early in the morning. For me, it's now it's 9. Okay, right. So now up to this point, is it very clear? Right. Okay, if you have any of the doubt in genetics, not like other things, here if you miss one part, if you do not understand one simple scenario, the next step will not be understood. Therefore, clear all the doubts, clear all the uncleared and understandable things then and there. Then it is very easy for genetics will be your easiest unit thereafter. Okay, right. I'll erase this one. No. We'll write this thing in the note and we'll move on to the next part, wasn't it? Mm, yeah, yeah, yeah. We do like that. So I'll share the screen of the tute. I hope all of you have downloaded it. Right. Genetics, number one, the scientific basis of Mendel's experiment, Mendel's heredity, or the Mendelianism, right? Right. Okay. The principles in heredity were first formulated by an Austrian Augustinian monk named Gregor Mendel. Is that Gregor Mendel? Gregor Mendel, right? Who is now considered the father of modern genetics. Considered the father of modern genetics. Father of modern genetics. Mendel discovered the basic principles of heredity. Basic principles of heredity by breeding garden peas in carefully planned experiments. Right. So he did all these experiments by using the garden peas, or you call it in Sinhala mungata කියලා කියන්නේ, right? So mungata වර්ග පාවිච්චි කරලා තමයි he did all these experiments. So garden pea, we learn how helpful was this garden pea for all these experiments, right? He has selected the very correct plant to do this experiment. Otherwise, he will be more confused at that time without the knowledge of genes or the chromosomes. He may have confused and will not succeed, but he has corrected the correct plan for these experiments also. His experiments were conducted decades before the concept of chromosomes. That is the most important part of that invention, right? The latter discovery of the chromosomes as the carriers of genetic units supported the Mendel's two basic laws of genetics, which are known as Mendelism. 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 Right. Okay. So, vocabulary in genetics. There are numerous heritable variations. Heritable or inheritable. Heritable variations. There are numerous heritable variations among individuals of a population. Among individuals of a population, such as brown, green, or blue eyes, or black, brown, or blonde hair in human population. So these are the heritable variations. A heritable feature that varies among individuals of a population, such as hair color or eye color, is called what? Is called a character. Is called a character in genetics. Character in genetics, right? These heritable variations of a character, these heritable variations of a character, heritable variations of a character in an organism, such as brown, blonde hair or blue, brown, or black eye in a human are called as what? So these variants are called as traits. They are called as traits. Traits. Right? These traits are transmitted from parents to offspring. From parents to offspring. Next point, from parents to offspring. Next point, observable traits. Observable traits. Observable traits of an organism is known as what? Phenotype. Phenotype. Observable traits of an organism is known as phenotype. Right. The Mendel has described about inheritable factor. He described inheritable factor. Mendel has described about inheritable factor in explaining his experimental results. In explaining his experimental results. These heritable factors are identified to be genes in modern genetics. Now we call them as genes in modern genetics. But at the era of Gregor Mendel, he named it as inheritable factor. He said that there is something inside the cell which gives rise to this phenotype. right? So the black hair can be observable, that is the phenotype, but to give that color to hair, there is something inside the cells that is the inheritable factor, right? So gene is the basic unit. Gene is the basic unit. Is the basic unit by which genetic information is passed from parents to offspring. So what do you mean by a gene? Gene means, Pute, all these things mean. Right. Now look at this one. You take one DNA, right? Usually the humans are diploid organisms. What do you mean by diploid, Pute? Oh, we have the pairs of chromosomes. How many pairs are there? There are 23 pairs of chromosomes in a human being, right? So one chromosome has come from the father, the other one has come from the mother, or else one is coming from the egg, the other one is coming from the sperm, right? Now, what do you mean by a gene? It means that it is a sequence of DNA. And you know, a chromosome means thousands and thousands, millions of nucleotides are arranged in a DNA molecule. So these are one DNA, right? So in a specific area, you have a sequence of DNA. Now, from here to here, you have a sequence of DNA, and this part is responsible for the, just assume that this part is responsible for the black hair, right, of a DNA. Now, this area of a DNA or an area of a chromosome is known as a gene. This is gene, right? So, it's a DNA sequence. You may have sometimes thousands of 10,000. So, number of nucleotides are arranged here, right? So, in the other chromosome, which are pairing at the cell division, these are paired only at the time of cell division, otherwise, they are haphazardly arranged, right? In the other chromosome, also, you have the specific DNA sequence which are responsible for the hair color. Hair color. So, for a specific gene that we are interesting or we are discussing, that gene is located in a specific place. There in another chromosome, in another place, you have a gene responsible for the eye color. Right? Likewise, in a chromosome, there are various number of genes are located. Here you have another gene, another gene, another gene, another gene, like that. Hundreds of genes are located in the same chromosome. right? So likewise, you take the 46 chromosomes. Each chromosome has various number of genes which are responsible for various number of characters, right? So all the traits are written in these chromosomes. That is the genotype. So when you take all the 46 chromosomes and when you take all the genes that are located in these chromosomes, the collection of these genes are called as the genotype. Understood? Right. Now, when you focus on one gene, now we are focusing on the character called as the hair color, right? So here we are focusing hair color. This is the character, right? So these genes are located in a specific chromosome in a specific place. That place is called as today. Now, this place in the chromosome is called as locus. Locus is the singular one, or else you call loci. So every gene is located in a chromosome in a specific place. That place is called as the locus. Right? Understood. Okay. So you have the homogeneous chromosomes. Homogeneous chromosomes means paired chromosomes, which one comes from the egg and the other one comes from the sperm. And both these homogeneous chromosomes, the same character, the gene which is responsible for the same character is located in the same place. Understood, right? The gene which is responsible for the same character, for example, hair color, right? The mother's hair color gene is located here. We'll assume that this is mother's chromosome and this is father's one, right? So mother's hair color gene is located here. That is the locus of the mother's chromosome. And this is the locus of the father's chromosome hair color, right? A gene, and it is located in the same place, right? So here, these genes which are responsible for the same character is called as alleles. Called as alleles. Now, what we are learning, Putin? We are learning the vocabulary, right? We are learning the technical terms used in genetics. Okay, very easy, right? Now, you know about the character, right? Hair color is the character. And here we learn about the gene. What do you mean by a gene, right? So, gene is a DNA sequence which is located in a specific place of a chromosome, right? If you take the two homologous chromosomes, that means the two pairing chromosomes, genes are located in the same place or the same locus. Locus. So you learn the word locus or the loci also, right? Genes and locus, right? Now, if you take the gene, the two homologous chromosomes are having the genes at the same place, and you have one gene in the mother's chromosome and the other one is from the father's chromosome. And these two genes are which is responsible for the same character. Again, I'm telling you, the genes which is responsible for the same character is called as alleles. Alleles. Okay, right. Now, sometimes these alleles can be equal. The mother is also having the gene which is responsible for black color, black hair, right? And the father is also having the gene responsible for black hair, black color, right? So therefore, the two genes are identical. two genes are identical. Okay. So if the two alleles are identical, you call it homozygous. Homozygous. If the two genes are equal, that means the one chromosome is having the gene responsible for black color. The other gene is also having the gene responsible for black color. Identical. There are some instances where these are not identical. For example, we'll think that the mother is having a gene responsible for black hair and the father is having a gene responsible for brown hair. Right? Just imagine one chromosome is having black hair gene at the same place. The other chromosome is having a brown gene. So then, are these two genes are identical or same? No, they are not identical, right? They are different. They are different. If the two alleles, if the alleles are different, then you call that status as heterozygous. Heterozygous. So if the two genes of the chromosomes or the alleles, if the alleles are identical, you call that organism or that status, he is a homozygous person. And if the two alleles are different, Pude, that is called as heterozygous. This is also possible. Right. Okay. Now, if the two alleles are identical, this one is also black color gene, this one is also black color gene, then the person is having a black hair. No doubt. Right? No complications. Both genes are responsible for black color, so the person is having black color. So alleles are same or homozygous. But in the case of heterozygous state, one chromosome is having black gene, the other one is having brown gene. So in this case, what will happen to the person? Now, the genotype is heterozygous. What could be the phenotype? Some of the hair is black color and some hair is brown color. Will it be like that? 50% of the hair black, 50% like zebra. No, it doesn't happen. Even the genotype is heterozygous. You show only one hair color. Right? Even at the state of the heterozygous, the person is having one phenotype. We'll say black color. So what has happened in that case? You have the black gene, you have the brown gene, both are there in the genotype or in the chromosomes, but the individual is having only one hair color. So that means, Pute, black color is dominant and it is the color that is shown in the individual. And the brown color is recessive and it doesn't show the color. So it is hidden. It does not exhibit any of its capabilities. Right? So in the heterozygous, right, only one allele, right? Only one allele is showing the phenotype, the other one is hidden, right? At the heterozygous state, right? So in the heterozygous state, the phenotype shown is called as the dominant phenotype. or dominant trait. Dominant trait. Or we first go here. The gene is black color. So black gene is the dominant gene. Dominant gene is the black. The other one, the brown gene, which do not exhibit any of its features at the heterozygous state, that is called as recessive gene. Recessive gene. So in this story, brown gene is recessive. Brown gene is recessive gene. So dominant gene black, recessive gene brown. So the trait black hair is dominant trait and brown hair is recessive trait. So you have the dominant gene and dominant trait and recessive gene and recessive trait. Okay, right. So alleles can have, again I am summarizing, you have a gene in a specific locus in homogeneous chromosomes. The same gene is located in the same place, right? So all the traits can be written in different types of genes. So alleles have different genes. So if the two genes are identical, that stage is called as homozygous. So you can have black and black, brown and brown, blonde and blonde, right? If the two genes are same, identical, that is homozygous. So if both genes are black, individual is black. If the both genes are brown, individual is having brown. If the both genes are blonde, individual is having blonde hair. No matter, no confusion. Simple. That is homozygous state. Homo means same, right? Homozygous. Right. So there are some instances you are having two different alleles. One is black, the other one is brown. One is brown, the other one is blonde. Right. Likewise, you can have different types of alleles. So if the alleles or the genes are different, that is called as heterozygous. In heterozygous state, only one trait, only one gene become dominant and the individual shows that color. That gene is called as dominant gene. In heterozygous state, the other gene which is hidden or which do not exhibit any of its features is known as recessive. Right? Angila තියෙන right? Recessive gene. And that trait or the color is the recessive trait. Okay? So these are the other words that you have to know in the genetics, right? Vocabulary. Okay, understood? Right. So usually, the other important thing beside this vocabulary part, now usually the outcome or the phenotype is decided by the gene plus environmental factors. Remember, this is an MCQ. Sometimes you can get an MCQ. The phenotype is 100% or solely decided by the genotype or the genes. Looks like correct, not, but not, right? Basically it is, but in biology science, in biology, Pute, the genetic is the most important genotype or the gene is the most important time, but it has some interaction with the environment also. For example, the person is having a fair skin according to the genotype, but he is living in a uh, sunny area and he is exposed to the intense sunlight, and you get additional deposition of the melanin, and the fair skin is converted to somewhat dark skin. So his gene is having a fair skin gene in the body, in the chromosome, but the environmental factors has adapted that thing to a darker skin. So phenotype today, remember phenotype is an interaction between the genotype and the environment. Right? Just keep that in mind and forget about that thing when you learn the genetics, right? Okay. So in genetics, right, what we call genes are responsible for the phenotype. Genotype will give rise the observable phenotype. Okay, right. Now you know about the dominant and the recessive traits as well. Okay, right. So another final thing about the recessive genes. Now assume that we call that brown gene is recessive here because in the existence of the black gene, it is not shown outside, right? Now just imagine this case, one chromosome is having the brown and the other one, the other allele also brown. So the both alleles are brown, but they are recessive. So what could be the phenotype of this individual? No doubt, no argument, that is brown color. So remember, even the recessive gene, when it is there in the homozygous state, even the recessive gene, when it is there in the homozygous state, that means both the genes are same, identical, then it shows that feature as the phenotype, right? So recessive genes cannot exhibit its color or the trait only when it is heterozygous, or else only when there is a dominant gene. When there is no dominant gene, the recessive gene exhibit without any hesitation his own phenotype. Okay, understood. Now you learn another set of words here. Any questions, Pute? Understood clearly? Okay, right. Shall we write this thing? Okay, it's very clear, right? Thank you. Right. Ah, right. We stopped at right. So Mendel has described about heritable factors. Mendel has described about heritable factors in explaining his experimental results. These heritable factors are identified to be genes in modern genetics. Gene is the basic unit by which genetic information is passed from parents to offspring, and it is a DNA sequence. It is a DNA sequence residing usually at a specific locus on a particular chromosome and contributes to the development of one or more traits. Development of one or more traits. One or more traits. By coding for specific proteins or peptides. by coding for specific proteins or peptides. Proteins or peptides. Right? That basic thing you know, right? So what happens? This gene will produce that responsible proteins in the protein synthesis. You know about the protein synthesis now, right? This gene is unwind before the protein synthesis, and that will produce the proteins which are responsible for brown color, proteins which are responsible for black color, proteins which are responsible for blonde color. So that is how the genotype is exhibiting the phenotype. Okay, right. Now there are alternative versions of genes. There are alternative versions of genes. There are alternative versions of genes which are called alleles. 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 There are alternative versions of genes which are called alleles. Alleles reside on the same locus. Alleles reside on the same locus. On the same locus of different chromosomes. In the same locus of different chromosomes. Alleles vary in their sequence of nucleotides, right? So, now when they are different, they have a variety of sequence of nucleotides. These changes can affect the function of the protein encoded by the gene and thus the phenotype of the organism. So if the DNA sequence are different in alleles, the protein that is produced can be different. Each diploid organisms, such as humans, has at least two copies of each gene. One copy in one homologous chromosome, the other one is in the other homologous chromosome. Each diploid organism has at least two copies of each gene residing on the chromosomes received by the two parents. Received by the two parents. Now you understand this story. These copies could be identical. These copies could be identical or could be different. These could be identical or could be different from one another. Right? From one another. The condition of having two identical alleles for a given gene, right? The condition of having two identical alleles for a given gene is known as what to think? If the two alleles, two genes, right? Two alleles of a gene is identical. Both are black and black, brown and brown, blonde and blonde. If the two alleles are identical, that state is known as homozygous state. Homozygous state. That condition is known as homozygous state. Alternatively, having two different alleles for a given gene is referred to as heterozygous state. Heterozygous state. So one gene is having two alleles. If the two alleles are same, that is homozygous state. And if the two alleles are different, that is heterozygous state. Right. Another part. Now, phenotype, this is what I told you. Phenotype is brought about by the interaction. Phenotype is brought about by the interaction between genotype, of the individual with its environment. With its environment. Right? So phenotype is brought about by the interaction between the genotype and the environment. MCQ point. Right? The genetic makeup or set of alleles of an organism, all the genetic makeup or the set of alleles of an organism is known as its genotype. It is known as its genotype. An individual's genotype could be either, right? So the genotype, now if you focus only one gene, the genotype can be heterozygous or the homozygous when you consider only one gene, right? So a person who is homozygous for hair color can be heterozygous for the eye color, right? Therefore, to decide whether the homozygous or heterozygous, we are defining it only for one gene of interest at a time, right? So, same individual can have heterozygous alleles and also the homozygous alleles for different genes. Understood that? Okay, right. So, uh, an individual's genotype could be either homozygous or heterozygous. Could be either homozygous or heterozygous with respect to a given gene. With respect to a given gene. Right. So at heterozygous state, that means two alleles are different. At heterozygous state, the allele which determines the organism's phenotype. Organism's phenotype. At heterozygous state, the allele which determines the organism's phenotype by masking the expression of the other allele is referred to as the dominant allele. Is referred to as the dominant allele. It should be the dominant allele, not the gene. Dominant allele. In the same gene, we have two alleles. So dominant allele and recessive allele. So again, that sentence. At heterozygous state, the allele which determines the organism's phenotype. So that allele is the one who is deciding what is the phenotype, whether it is black or brown, right? Organism's phenotype by masking the expression of the other allele. The other allele's expression is masked, right? Is referred to as the dominant allele. Dominant allele. The trait produced by the dominant allele is known as the dominant trait. It is known as the dominant trait. Known as the dominant trait. The allele which does not exhibit any noticeable effect on the organism's phenotype, where when at heterozygous state is referred to as the recessive allele. Is referred to as the recessive allele. Recessive allele. The trait hidden on the recessive allele, the trait hidden on the recessive allele is the recessive trait. The trait hidden on the recessive allele is the recessive trait. recessive trait. However, they express their trait when they exist in the recessive allele also pute express their trait, right, when they are in homozygous state. However, they express their trait when they exist in homozygous state. Homozygous state. Right. Okay. Understood. Any questions up to this point, Pute? There is a question. Fish. So, do alleles always stay on same locus? What about the heterogeneous chromosomes? Yes, yes. Alleles are placed on the same place. That means parallel, right? Now you take the homologous chromosomes. Now don't get confused with these two words. Homologous means Pute, homologous chromosomes means the two chromosomes that comes, one comes from the father, the other one comes from the mother, right? Now you take the human beings are having 46 chromosomes. Out of these 46 chromosomes, Pute, 23 has come from the sperm, the other 23 has come from the ovum or the egg, right? So when you make the zygote, all these 23 chromosomes from the sperm and the other 23 from the egg is mixed, right? Now, there you have the these two the 23 sets, 23 chromosome, one set in the sperm, the other set in the egg, are contributing or responsible for the same traits, right? So each chromosome has a pairing another chromosome in the other set, right? So likewise, that pairing other chromosome is called as the homologous chromosomes, right? So you assume that this is the 13th chromosome of the sperm and this is the 13th of the egg. So those two chromosomes are the homologous chromosomes. In these homologous chromosomes, in the same place, the alleles are located. Either it is heterozygous, either it is homozygous, the two alleles are located in the same place. Okay, Krish? Right? So they are located in the same place. If the two alleles are same, you call it homozygous state. If the two alleles are different, you call it heterozygous state. That's all. Okay, good. Right. Any other questions, Pute, before we move on to the next part? Still, Pute, we are learning the technical terms in genetics, vocabulary. Still, we are learning the vocabulary. Now, you know a lot of things starting from the heritable factors. Okay. Right. Shall we move on? Okay. We'll do the class for 2 hours and we'll take a break. Some students said that they have a class in the morning, so you can uh, leave and then later you go through the recording. Right. So this week, the Friday also we are having the class. Right. Friday night we'll have the class as usual. Yesterday I had a unexpected thing in this in my apartment in the Malaysia. Right. That is why I couldn't hold the class yesterday. Right. But usually 2026 class we are going to have the class twice a week. So that is how we are going to cover the things. And also by the time we'll do some full papers also with the beginning of the next month, right? So that will cover all the things. Okay, so don't worry. Right. Another two things that is important about the Gregor Mendel today, right? Why his experiments about the genetics were so successful, even at an era where people don't know about the chromosomes or genes or nothing is known. At that time also, Mendel's experiments were so successful because of two things. So one thing, he traced or he was focusing the traits which are very distinct to each other. He was using or tracking distinct variations. Right? That means you have very contrasting phenotypes, distinct or contrasting phenotypes. For example, today he focused about the flower color of the garden pea. Some flowers are purple in some plants, and in some plants they are having white color flowers. So very contrasting. There is no in between one. Some are purple, some are white. Very contrasting, right? So he focused on these contrasting phenotypes only. One reason for his successive rate in the experiments. The other thing is he took for the initiation of the experiments, he took the pure breeding purple, pure breeding traits, pure breeding purple, pure breeding white. He took the pure breeding lights, or you call it true breeding plants as the parent plants. So, what do you mean by true breeding today or the pure breeding? Now, to start the experiment, now when you go to the environment today, when you go to the garden pea plant, right? And there is another question. So then in heterozygous one allele is located in different locus, or it is the different allele in the different locus, or both. In heterozygous one, one alleles are located in the different locus, so different alleles in the different loci for both. What you are asking, Gaggy? Now you have the now I'll take this is a one chromosome, and you have the take another color. Right. These are the two homologous chromosomes, right? So assume that we have the allele for hair color here in one chromosome and allele for the hair color in the other chromosome. So this is, assume this is brown allele and this is black allele. So it is heterozygous, right? So they are located in the same locus of the chromosomes, homologous chromosomes. That means same locus means at the same place. It is not now the brown allele is located in the father's one in this place, and the black allele is not placed somewhere here. It is not that. Understood? Same place means at the same level. At the same level. That is why you call it same locus. At the same level, right? Either it is homozygous, either it is heterozygous. The two alleles are located in the same locus of the homologous chromosomes, right? So, irrespective of what is the allele responsible for the same gene, for the hair color. Now, here, what we are focusing is hair color gene. Gene එකේ නම මොකද්ද පුතේ? hair color. Right? ඒ hair color කියන gene එකේ variations දෙකක් තියෙනවා. alleles දෙකක් තියෙනවා. Brown, black දෙකක් නෙමෙයි තුනක් තියෙනවා. Right? You have three alleles. Brown allele, black allele and blonde allele. All these, whatever the two alleles out of these three are located in the same place of the two chromosomes. Okay, irrespective whether if the two alleles are same, you call it homozygous. If the two alleles are different, you call it heterozygous. That's all. But they are located at the same level of the two chromosomes. Okay. Right. Good, good. Ask any question, Pute. Okay. What about XY chromosomes? Their lengths are not same, so are corresponding to alleles are absent for the same. Right. That is an exception. Right. So we are talking about that thing at the later, in the middle part of the genetics. That is an exception. That is why I did not confuse you at the same time. Right. There are some exceptions in X and Y. So those are the sex chromosomes. In sex chromosomes, Pute, in that case, you have different the different locus, right? In only in some things, right? But in the XY also there are some genes which are located in the same locus also, but there are some genes which are located in different places, right? That is an exception, Krish. That is an exception. You are true, right? But at this level, I did not confuse you, right? So this is the normal theory for majority of the genes. 99% of the genes are located in the same locus. But there are some exceptions. Example is the sex hormones. Here the X, the father gives you X or the Y and mother gives you the X. So that will, the genes, the lengths are different. So there can be the locus or the loci can be different in some genes only. Okay. Right. Right. Good, good, good. Right. So right. So, what we were discussing, the Mendel was so successful because of two things that he focused only on the contrasting phenotype, right? The purple, white, long and short, tall and dwarf, right? Round and wrinkled. Likewise, the character that he focused had very contrasting traits, right? For example, if you take do an experiment among the human beings, you take short and the tall. Can you take that character? Some people are short, some people are tall, but is these traits are contrasting? No. පිට කියන්න බෑනේ මූ කොටයි මූසයි right උස කියන එක that is a you can't define, you can draw a clear line in between these two. අඩි 5 කෙනාට the person who is having 5 feet can be defined as tall compared to a person who is having 3.5 feet. And he can be a shorter one compared to a person who is 6 feet tall guy like me. Right? So they are not contrasting traits. But Mendel took very contrasting phenotypes, so there were no any confusion. The second one, he took pure breeding ones to start the experiment. What do you mean by this pure breeding one means? If you want to do a cross in between the purple flower garden pea and white flower garden pea, he just did not took a purple flower seed and a white flower seed. What he did before he started the experiment, he took the purple color seeds, and it crossed with the purple, and in the next generation, some were purple and some were white. He neglected this white part. These people, again, the purple plants self-pollinated with purple. Again, majority purple. Very few white were there. Neglected the white. This one self-pollinated again. So likewise, for few generation, he crossed only the purple, and got the pure breeding line of purple. If you want to take white, they did this with white, and got the first generation as purple and white. So he neglected purple. White color was bred with again white, and got the pure breeding white again. Right. So these are the two successful steps that the Gregor Mendel has taken at that era. That is why his experiments were so successful and he became the father of genetics, right? So remember two things. He focused or he traced or tracked only contrasting phenotypes, and also for the start of his experiments, before the experiment, he selected the pure breeding traits of both the traits, either purple or the white. For each and every experiment, he took only the pure breeding plants as the parent plants. Okay, so those are the two things. And they, right. Now, very important part. Now we are moving on. Right, still we are discussing the vocabulary, huh? Okay. Right. Now I told you that Mendel selected the pure breeding lines for the experiments. And he took white flower garden pea, right? And these plants were cross-pollinated with purple color flowering garden pea plants. These are pure breeding lines or true breeding lines that he took after several generations of crossing each other. So this is true breeding white flower plants were crossed with true breeding purple flower garden peas. Right? Now, This is known as hybridization, right? Hybridization. Now you knew this hybrid word when the hybrid vehicles come, no? Usually the vehicles were running with the fuel, either petrol or the diesel, and later the technology comes as sometimes the car is driving with the fuel, and sometimes it is driven by the charged battery. So two contrasting thing is contributing to the drive of a vehicle, battery charge and the fuel combustion, right? So that is hybrid. So before the vehicles are made in hybrid, Gregor Mendel did the hybridization. So the hybridization word comes in genetics. So what is this hybridization, right? Contrasting one trait is cross-matched with the contrasting other trait. So the purple color plants were cross-matched with the white color garden pea. That is that cross-match, that pollination is called as hybridization, hybrid color, right? Two contrasting things, hybridization, right? So here, the first generation to start the experiment that you took white flowering garden pea and the purple flowering garden pea. So this generation is called as the parent generation, that he is focusing on this generation. So this one is called as the P generation. P generation. That means parent. P can parent. Parent generation. Right? So then what happens with the pollination? You get the seeds of these plants, and these seeds which were occurring in the white pea plant one and the purple garden pea plant, all these seeds were again uh, grown as the second generation, right? And that one you get again. So Mendel observed that all the next generation, right, which are grown from the seeds were having only the purple flower, right? Purple flower, right? So the next generation, which he took from the parent generation, is called as the F1 generation. This is called as the F1 generation. F1 generation. So parent generation is the generation that you start the experiment. The next generation that you got from the parent, from pollinating the parents, is known as the F1 generation. F word coming from the Latin word filial. 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 Right? Filial means putte son, puta. Right? So the parent generation, the first filial generation is F1 generation. And he proceed to another step. He cross-matched F1 with F1, self-pollination or the cross-pollination only from this generation that you got from the parents. Same plants were cross-matched each other. That is, you call self-pollination or cross-pollination of these things. So purple, again, cross-matched with purple flower of the F1 generation. And they got another set of generation, that is the third generation. First generation is the parent generation. Second generation is the F1 generation. Third generation is the F2 generation. And there you got purple color plants plus white color plants as well. Right? So that is the F2 generation. F2 generation. Right? So P generation, parent, if P generation, F1 generation, and F2 generation. First generation is the parent generation. Second generation is the F1 generation. Third generation is the F2 generation. Okay? Understood? Right. So, another thing. Right? This is the basic structure of the genetics experiments that he did in the genetic experiments. Okay? So, what are the things? Again, I am summarizing. William Mendel, right? He chose very contrasting traits. And before he started the experiment, he took the PO lines, PO breeding lines or true breeding lines to start the experiment. This is how he started the experiment. So he took PO breeding contrasting traits and cross-matched. So that generation is considered as the parent generation. So every experiment he did for twice, or he went for two new generations. So altogether, all his experiments are having three generations. Parent generation, F1 generation, and F2 generation. Right? Now you know about the gene and the alleles. Assume that these garden pea plants, Pute, having a gene which is responsible for the color of the flower. Garden pea plants is having a gene which is responsible for the color of the flower. So one gene. And two alleles are there here. One allele is for the white color, the other one is for the purple color. Right? So they are located in the same locus. Okay, right. Now, look at this one. What has happened in this experiment? In the parent generation, Pute, you are having the white color flower plants and also the purple color flower plants. And in F1 generation or the first generation, all the plants, 100% of the F1 generation were purple color ones. Purple color ones. So what do you understand by that scenario? In the gene is the color of the flower. Gene is the color of the flower. And alleles are, two alleles are there. One is responsible for white, one is responsible for the purple. And in the first generation, you had only purple color. So in that case, what do you think about the dominant allele and the recessive allele? Now tell me, what is the dominant allele in this case? Dominant allele is purple, isn't it? And the white is recessive. That is why in the first generation, you had only the purple color. When you mix the two alleles in the two chromosomes, one is the purple allele, the other one is white allele. The white allele is not exhibited. It is hidden. It is recessive. When the two alleles are there, or in the heterozygous state, only the purple color is exhibited. So in that case, purple color is having the dominant allele. It is the dominant allele, right? So we'll take purple flower, the genotype, we'll take capital A. Right? Now in genetics, Pute, to demarcate, to denote the dominant allele, you always use a capital letter. If it is a recessive allele of the same gene, you use the same letter but in simple version. Right? So these are pure breeding ones, no? So purple flower means that genotype, the both alleles are purple. Capital A, capital A. Understood? Right. Because he took only the pure breeding ones. Pure breeding means pure, the both genes are same. Right? Homozygous. These are homozygous. Right? So he took white color ones. So their genotype should be simply. Why? It is recessive, no? But they are pure breeding, therefore both are simple A, recessive variants. So simple A, capital A, crossed each other, right? Hybridization done. In this step, hybridization done, and you get the F1 generation. What could be the F1 generation genotype? All are purple, but in this generation, one gene is coming from this plant, the other gene is coming from this plant. So here the genotype is capital A, simple A, heterozygous. This is homozygous white, homozygous purple. If one generation you got all the plants in purple, but one allele came from one plant, the other allele came from this plant. So the genotype should be capital A, simple A. And this is a heterozygous state. So in this heterozygous state, you have the dominant allele and the recessive allele. So the phenotype is decided only by the dominant allele. So dominant allele is capital A, which is responsible for the purple color. Therefore, 100% of the F1 generation, all the plants were purple color. Right? So, next step, what he did, F1 plant pollinated with another F1 plant. So, capital A, simple A, pollinated or crossed with another capital A, simple A. Right. Now, this step, Pute, right? You are focusing only one gene. What is that gene? You are focusing only the color of the flower. Now, you are crossing two plants which are heterozygous for that color. Right? Capital A, simple A plant, purple color, is crossed with another heterozygous capital A, simple A plant, purple color. Now, this cross is called as monohybrid. Monohybrid cross. Why? Mono means Pute only one. You are focusing only one gene. The plant is having all the genes, but at this experiment, you are focusing only one. Now, pure breeding contrasting two plants are crossing. What do you call this one? That is the hybridization. So, in the hybridization, as the first generation of filial, F1 generation, you get the heterozygous plants, right? So this heterozygous plant is, in this experiment, you are identifying them as a monohybrid. This one plant is known as monohybrid plant. So capital A, simple A means a monohybrid plant. This is a monohybrid. So monohybrid is crossed with another monohybrid. Why you call it monohybrid? It is hybridization done, but you are focusing only one gene, mono, one feature. You are focusing only the flower color. So monohybrid plant. Monohybrid plant means it is an individual who is having dominant allele and the recessive allele for the for the gene that we are focusing. Gene of interest. එකට අපි කියනවා gene of interest. ඒ වෙලාව අපි බලන්නේ මොන gene එකද? Gene of interest. What we are looking at that time. At one time you are focusing only one trait, one character. Sorry, one character. That is the color of the alleles. So, individual or a plant who is having heterozygous alleles for the focusing gene is called as a monohybrid. So, second step, monohybrid crosses with another monohybrid, that is a monohybrid cross. So, F1 crosses with F1 means monohybrid cross. Okay. In a monohybrid cross, as the result, right, F2 generation is there. You get the F2 generation, right? So, in the F2 generation, what has happened to them? You are having purple flower plants and also the white flower parts. Now, in this F1, you had only the purple and purple. You are crossing only the purple and purple, but in the seeds, F2 generation, you get the purple plants and the white plants as well. Right? So, next step, we'll see how this has happened. Right? So, usually, right, this is known as monohybrid cross. So, Mendel Pute, in this whole session, we are going to learn about two theories of Gregor Mendel. Those theories or laws, they are laws. Law means the law. It is not like evolution, theories of evolution. Evolution, we discussed about theories. You can't prove that 100%. Therefore, theories of evolution, Lamarck theory, all these theories came as they are known as theory. Darwin's theory, right? Theory. But here you have the laws. Law means law. You can't change it, right? Not in Sri Lanka. Sri Lanka law is changing according to the type of government that you have. You forget about that thing, right? Genetics. This is law. So Mendel's presented two laws. The first law of inheritance, second law of inheritance. Now, first law of inheritance, he presented by using this monohybrid cross. Right? Just now, still we are learning the vocabulary now. So, learn what is hybridization, what is monohybrid cross. Okay, right. Then, when we will write this part and we'll go to the next part. Then we are going to learn about what happens when you focus two genes, two features. For example, color of the flower as well as the shape of the seed. So when you are focusing two characters at the same time, another set of words comes. Right? Up to this point, is it clear for you, Pute? Okay. Sorry that I am muted. Okay, thank you. Right. So you are asking Situmli, even F2 generation purple plants are showed as capital A, simple A. No, no, no. It's not. I'll explain it. That is different, right? So that we will go for that expansion in the next step. Still at this point, you just know this is hybridization. Pollinating two pure lines is called as hybridization, right? Pollinating heterozygous two plants, monohybrid cross. One heterozygous plant is known as monohybrid. So here, some are, for the time being, Pute, some of the purple color ones can be capital A ones. And there are some ones, capital A, simple A ones. White ones are for sure simple A and simple A. How you get homozygous capital A, how you get heterozygous capital A and simple A, we'll discuss by drawing a box, right? So here the purple color ones are either homozygous or either heterozygous. White ones are anyway homozygous. Okay, I'll explain how it comes in the next step. Still, I am just teaching you the technical terms. Okay, right. Right. We'll write this one and then move on to the other type of experiment which you are focusing two characters at the same time. This is monohybrid cross. Okay, right. Right. The Mendel tracked only those characters that occurred in two distinct contrasting phenotypic forms, such as tall stem versus short stem length, or purple flower color versus white flower color. That is one reason that why the Mendel was successful. And such traits are referred to as contrasting traits. So he focused only on contrasting traits. Contrasting traits. Then the other reason, Mendel used only the pure breeding. He used only the pure breeding, sometimes called as true breeding varieties for his experiments. Pure breeding plants are obtained by, how you get the pure breeding plants? By self-pollinating, over many generations. By self-pollinating over many generations. Producing only the same variety as the parent plant. So he took only the pure breeding ones as the parent generation, P generation. Right. These uniform lines produced from self-fertilization of pure breeding varieties over many generations are called pure lines. These are pure lines, right? Are called as pure lines. So pure lines are anyway pute homozygous, either for the dominant allele, either for the recessive allele, they are homozygous pure lines. So no doubt about this white color one, it is simple and simple A, pure line. No doubt about this purple flower, it is capital A, pure line. You don't have the capital A and simple A lines here as the parent generation because he took only the pure lines. During his experiments, Mendel cross-pollinated pure breeding garden pea plant varieties which shows contrasting traits. For example, purple flower plants were crossbred with white flowered plants. Now, mating or crossing of two pure breeding varieties with contrasting traits is called hybridization. This is called as hybridization. Hybridization. Mating or crossing of two pure breeding varieties with contrasting traits is called hybridization. Hybridization. Parental generation is referred to as the P generation, capital P. Use the capital P. P generation. Parental generation is referred to as the P generation or the parental generation. Plant progeny. Progeny means the resultant ones, right? The plant progeny resulted from these hybridization events are referred to as F1 generation. F1 generation. F1 generation. First filial generation. The word filial comes from the Latin word for the son. F1 generation. The progeny that results from the self or cross-pollination between these F1 generation plants are known as the F2 generation. So the progeny got from the cross-match of the F1 is called as the F2. So progeny from the parent generation is the F1. Progeny from the F1 and F1 is the F2 generation. So between these F1 generation plants are known as F2 generation of or the within bracket second filial generation. Right. The other next point. An organism. So focus on this one. And when you are writing this sentence, focus on this one. The F1 generation. Focus on F1 generation. An organism that is heterozygous. An organism that is heterozygous with respect to an organism that is heterozygous with respect to a single gene. You are focusing only a single gene. An organism that is heterozygous with respect to a single gene of interest resulting from a cross between parents having homozygous conditions. A cross between parents having homozygous condition. Now, focus on P generation. Now we are writing P generation. Right? Parents having homozygous conditions for different alleles of one specific gene. Is referred to as a monohybrid. So F1 එක plant එක individual කෙනෙකුට අපි කියනවා monohybrid. You call one individual in the F1 generation as monohybrid. Right? So that is what we wrote. An organism that is heterozygous with respect to a single gene of interest resulting from a cross between a parent having homozygous conditions for different alleles of one specific gene is referred to as the monohybrid. Right? So breeding experiment conducted between two organisms with heterozygous conditions for a specific character is referred to as monohybrid cross. So crossing F1 with another F1 is a monohybrid cross. So result of that monohybrid cross is the F2 generation. and you stop the experiment from this one. Right? Very interesting. Very clear. P generation, PO lines, homozygous. That is hybridization. You get the F1 generation, right? That should be heterozygous because P generation was homozygous for both alleles. So F1 generation is heterozygous, and that F1 generation, one individual is known as monohybrid. So you are crossing monohybrid with another monohybrid, that is a monohybrid cross. So F1 generation, here what happens in this step, you have the monohybrid cross. So result of that monohybrid cross is the F2 generation here, F2 generation. Right. Okay. Any questions put up to now? Now only one final point is there in the vocabulary. Yeah? Now we are at the end of the learning vocabulary. Now if you know these things very clearly, if you understood this very clearly, genetics is very easy. Right? Then maybe I had the theme. Okay. Right? Okay. So, and assume a scenario here. Now, in the first instance, Pute, we focused only one character. Now, Mendel did another set of experiments. There, he focused two characters at the same time. Two characters at the same time. You have the P generation, right? In the P generation, Putin, he is having purple color flowers and round seeds. Pure breeding ones, right? And they were crossed with white flower plants, and they are having wrinkled seeds. Right? In round seed means the seed is like this, round and smooth. Here, the outer covering is wrinkled, right? So, purple flowers, round seeds. Now you are focusing two characters. You are focusing the color of the flower as well as the shape of the seed, right? So you at the same time you are focusing two characters. Purple flowers, round seeds. White flower, wrinkled seeds. Now this is the P generation, and you got the F1 generation as purple flower, round seeds. So this is the F1 generation. F1 generation. Right? So, next step is crossing the F1 with another F1. Purple, round, another plant. F1 crossed with the F1. So the progeny you get is the F2 generation, and there you had different types of variations. Purple, round ones came, purple, wrinkled ones came, white, round ones came, and white, wrinkled ones came. Forget about this result, right? We'll work on it. This generation is F2 generation. F2 generation. Now, what is the difference here compared to the previous one, Pute? Here we are focusing two characters, right? So here you have the pioline, purple flower, round seeds. So that means you are having pioline means this is the color of the flower, capital A, capital A. Round seeds, as you get 100% of the F1 as the shape of the seed as round, that means round shape is dominant allele. Wrinkled seed is recessive allele. Why? F1, 100% all the plants were purple color and the round seed. So round shape is dominant. So purple flower, genotype is capital A, capital A. Round seed, genotype is capital R, capital R. This is the genotype of parent generation because he took only the PO lines. He took here again the PO lines. So white color means simple lay, simple lay. Wrinkled seed means this is capital R, no? So the recessive one, simple R, simple R. This is the genotype of white color flowers with wrinkled seed. This is the genotype of purple color flower with round shaped seeds. Now, if one generation came as 100% purple round color, that means the genotype is put here capital A, simple A, Y, heterozygous. One gene comes from here, allele comes from here, the other allele comes from here. Capital A, simple A. Shape of the seed, one comes from here, capital R. The other allele comes from here, simple R. So 100% of the F1 generation is capital A, simple A, capital R, simple R. Heterozygous hybrids. This is hybridization. And what is this? In the previous one, we called this is a monohybrid. That is the result of or progeny of hybridization. Now here the hybridization is happening, but you are focusing two characters. Therefore, this progeny, this plant is called as one plant, one individual is called as dihybrid. This is known as a dihybrid. Dihybrid. So, to get the F2 generation, Pute, dihybrid are crossed with another dihybrid, self-pollinated or cross-pollinated, only the F1 generation. So dihybrid crosses with another dihybrid. That means what you do here in the F1 generation, right? You do a dihybrid cross. Now here what you do is dihybrid cross. As a result of dihybrid cross here, you get the F2 generation. Okay, understood? Right. Now, previous one, only very, very simple. You are focusing only one character. So, F1 is a monohybrid. F1 into F1 is a monohybrid cross. Here, you are focusing two characters. So, F1 is a dihybrid. And what you get from the dihybrid cross, what you do here is called as a dihybrid cross. You cross or pollinating one dihybrid with another dihybrid. So that is a dihybrid cross and you get the F2 generation. Okay, that is the two concepts you learned. What is monohybrid cross and what is dihybrid cross? Very, very easy. Don't afraid of these words. Now you know every minute of this, every minute of these words. Understood clearly? දැන් බය වෙන්නේ නැහැ නේ monohybrid cross කියලා තිබ්බහම dihybrid cross තිබ්බහම very simple. Monohybrid cross, you are focusing only one character, right? And it is heterozygous. Monohybrid is a heterozygous always. Dihybrid, you are focusing two characters at the same time and it is heterozygous. Again, heterozygous. So monohybrid or dihybrid always heterozygous. Monohybrid, heterozygous for one character. Dihybrid, heterozygous for two characters. That's all. Dihybrid cross. Okay, clear? Right. Shall we write this thing? If you have anything not clear, please ask. Right. Right. The next point. An organism that is heterozygous. An organism that is heterozygous. An organism that is heterozygous with respect to two genes. An organism that is heterozygous with respect to two genes of interest resulting from a cross between parents having homozygous conditions. Two genes of interest resulting from a cross between parents having homozygous conditions. Parents having homozygous conditions. Parents having homozygous conditions for different alleles. Homozygous conditions for different alleles of two specific genes. Homozygous conditions for different alleles of two specific genes is referred to as a dihybrid. Is referred to as a dihybrid. Dihybrid. Now, breeding an organism having sorry, is referred to as a dihybrid. So here, F1 is a dihybrid, right? Breeding experiment conducted between two organisms with heterozygous conditions for two specific characters is referred to as a dihybrid cross. Dihybrid cross. It's a dihybrid cross. Okay? Right. Hope you are clear up to that point. So still we are learning the vocabulary. Now vocabulary is almost all the things are done. We have to learn only another one word. Right? If you have any questions, Putri, you can just ask. Right. Okay. No questions. Good. Now, one last point, Putri. Right. Now you take the dominant allele, Putri. Dominant trait. You take the dominant trait. Right. And so here in this example, the dominant trait of the color of the flower is purple, isn't it? So this is purple, right? Now, purple is the phenotype, purple color, right? Purple color is the phenotype or what you see, observable. And the genotype can be two things, okay? If it is a pure line one, it should be a homozygous one, capital A, capital A. But in the environment, you can see some of the purple flowering garden piece. You don't know what is the genotype. It can be a capital A and simple A also. Right? Now I give you a purple color flower garden pea plant. And I ask you, what is the genotype of you? You don't know anything. You can say it is the phenotype is purple color. But what you can say is genotype is not simple A and simple A. Make a simple A උනා නම් මොකද වෙන්නේ? What happens if the genotype is simple A and simple A? Then it should be a white color one. And I will give you a white color plant. Phenotype is white color and I ask you the genotype. Can you tell me the genotype here? Yes, obvious. It should be simple A and simple A. Otherwise, the white color phenotype is not there. Right? For having, if you are given the recessive trait and ask the genotype, you don't have any question. The genotype is homozygous. But if you are given a dominant phenotype plant, and if you were asked what is the genotype, you can't give a clear answer because it can be a homozygous capital A and simple A or else heterozygous capital A and simple A. Both genotype will give you the phenotype of purple color flowers. Now, how to resolve this problem? Now, you are given with the purple color plant and ask you what is the genotype. So, what you can do? You have to plan an experiment to find out this thing. You plan an experiment. What is the experiment, Putin? What you do here, right? You don't know whether it is capital A or simple A. What you can do is you cross-pollinate this plant. Now, this is the one that we don't know. Right? You don't know the genotype of this one. We don't know whether it is capital A or capital A and simple A. So what you do, you cross-pollinate it with the homozygous recessive trait. That means white plant, white color flower. And look for the F1 generation. Right? Now, white flower genotype is simple A and simple A. Very simple, right? So, in the F1 generation, Pute, if you have only the purple color ones, 100% purple color plants are there in the progeny, in the F1. So, we are looking for this one, no? What is this plant? We are trying to find out the genotype of this one. So if all the plants in the F1 generation are purple, that means what is the genotype of this one? Capital A, capital A. Why? Only capital A has come from that plant. And anyway, simple A is coming from this one. So to find out what is the genotype of this purple color flower plant, what you do is you cross match it with the white flower or recessive homozygous one. And if 100% of the F1 generation are purple only, that means this one is a capital A, capital A. That means homozygous dominant alleles are there. Just imagine you get some plants in the F1 generation, most of them are purple, right? And you get white flower plants also. In the F1 generation, if you get the white flower plants also, that means what is the genotype of this one? So you have got simple A from this one also. That is why the white color plants were there. Right? So that means the genotype of this plant is capital A, simple A. Right? So in the second instance, if you get the white and purple, this is the genotype. If you get only the purple color in the first answer, right, then the genotype is uh, capital A and capital A, homozygous. Understood? So what we did here, we wanted to find out the genotype of a dominant trait. For that, what we did was we crossed it with the homozygous recessive trait and looked for observed the F1 generation. And by observing the F1 generation, we decided the genotype of this plant. These type of experiments, these type of process are called as test cross. This is a test cross. Why? This is done to test what is the genotype of this plant. Okay, right. That is the end of vocabulary. Okay, right. So you know about three types of crosses: monohybrid cross, dihybrid cross, test cross. That's all. Okay, right. So we'll fill up that part also. Right. So breeding an organism having unknown genotype. Breeding an organism having unknown genotype for a specific dominant trait. For a specific dominant trait. Specific dominant trait. With an organism having homozygous recessive condition. So you are crossing with an organism having homozygous recessive condition. Homozygous recessive condition. Recessive condition. For the same specific trait is called a test cross. Is called a test cross. Test cross. Right. This is usually done to reveal the unknown genotypes of a specific dominant traits in an organism. Right. So what you do is, why you do it to reveal the genotype of a unknown dominant trait. Reveal the genotype. Okay, right. Then we have to move on with the genetics proper theories. Okay, up to now, you learned the very important things in the genetics. That means you have learned all the vocabularies, and some of the theories are covered when you learn the vocabulary also. You know all the things about uh, a trait, what is a character, what is a trait, and what is a phenotype. Know about the heritable factors, right? And you know about the locus and the loci, and you know about the alleles, dominant allele, recessive allele. You know about the dominant trait, recessive trait, and you know about the homozygous state and the heterozygous state, and also you know about the crosses, monohybrid cross, dihybrid cross, and the test cross. And you know about the P generation, F1 generation, and F2 generation. Okay, right? If you understand all these things, now very easy to proceed with the complex theories. Any questions, Pute? Right. So if you don't have any questions, we'll move on to the next part and shall we take a 15 minutes break? Right now we discussed for about 2.5 hours, right? So we'll take a 15-minute break in Sri Lankan time now. It is 8:30. 8. So we'll start around 8:45, right? We'll take a 15 minutes break. By the time, if you have any questions, you just drop a message, the chat box. Okay, right. So uh, we'll have a short break of 15 minutes and we'll meet again at 8:45. Okay. Right. Okay. Right. Now you know all the words or the technical terms in genetics, right? Now you are experts in vocabulary of genetics, right? Right. Now I told you that Mendel presented two laws in genetics, first law and the second law. Now we'll move on to see what is this first law. Very, very simple. Now most of the things now you know. Uh, right. Okay. Now here in the first set of experiments done by the Gregor Mendel, he focused, I told you that he focused contrasting traits of the same character. At the same time, he started with pure lines to start the parent generation, right? So in the first set of experiments, he focused only one character, right? And he did some experiments. Okay. Now you don't know about the genotype or the heritable factor there, right? You know only about the phenotype. Gregor Mendel can see. Now we'll take, for example, he focused only about the color of the flower. Color of the flower. He focused one character, right? So in the garden key plants, he saw purple flowers are there, white flowers are there. Both these colors are there in the phenotype, right? So he got pure lines of the purple color flowers and the pure lines of white color flowers. And he started the first step of the T generation, parent generation. What he did was purple color plants were crossed with white color plants, white color flowers. This is his P generation, right? P generation, right? So he observed that all the progeny, all the plants he got in the F1 generation were purple flower plants, purple color plants. So this is F1 generation of Mendel's experiments. Purple color plants, right? So 100% purple color plants. And then he did the monohybrid cross. He self-pollinated or cross-pollinated F1 plants into F1, right? F1 cross with again F1 purple plants. And he got the progeny. He got majority of the plants were having purple color flowers and also some are having white color flowers as well, right? So he says he took 705 plants in the F1 generation, cross matched, right? Crossed. And he got 705 purple color plants from the F1 generation and about 224 plants in white color flowering plants, right? So that is what he got. So it says, now you can see about the proportion between, now this is the F2 generation. F2 generation. This is the pure experiment that he did, and this is the result that he got. Right? So if you compare the proportion here, there is 3 to 1 proportion is there. 3 to 1 proportion. Now he wanted to elaborate, explain how this happened. Right? So he can notice or he can observe only the phenotype. No, he doesn't know about chromosome. He doesn't know about a gene. So this is the phenotype that he observed during his experiment. Purple color plants cross-matched with white color plants. These are purebred ones. And F1 was 100% purple flowers. F1 crossed with F1, this is a monohybrid one, cross, and in the F2, you got the purple and the white flowers into 3 to 1 proportion. 3 to 1 proportion, right? Now he wanted to explain how this happened. For that, he used heritable factor, right? Now the purple and white crossed today. I hope you can hear me. Right. Right? And got 100% purple. That means purple color is the dominant allele. So here the heritable factor of purple color plant here is capital A, capital A. Genotype is capital A, capital A. White color is recessive, therefore this is simple A because they are pure lines or homozygous ones. Homozygous purple, homozygous white, therefore capital A, simple A and simple A. And 100% of the F1 should be one gene comes from here, heritable factor comes from here as capital A, the other one comes from here as simple A. So 100% of all the plants in the F1 generation, the genotype is capital A and simple A. And they cross-matched, and the monohybrid cross is done, and you get the F2 generation as 3 of purple plants, you get only 1 white plant. So the proportion is 3 to 1. So how this happened, that is what he explained in his presentation. Right? For that, he used a square called as Punnett square. Right? So, this is P generation. This is F1. This is F2. Right? So what he said was, now when you are crossing the parents, purple plant and the white plant, what is crossing, Pute? A sperm will cross with the egg. So the purple flower plant produces capital A sperms as well as capital A eggs. White color plant produces simple A sperms as well as simple A eggs. So purple color egg can cross with the white color sperm. Again, purple color sperm can cross with the white color egg. Both ways can happen random. Right? So finally you got this one. Right? So what Brigham Mendel told was when the purple color plant is producing eggs and the sperms, these heritable factors separate, and go into the gametes. Gametes means eggs and the sperms separately. So this one capital A separately goes to eggs, and the other A goes to sperms, right? And here also, the simple A separate, and one gene goes to the sperms, the other one goes to the eggs. And the theory he said was heritable factors, all the heritable factors separates and go into the gametes, either eggs, either sperms, go into gametes independently, right? It is randomly happening. So separation of heritable factors is the theory that he told as the first law. So what is the first law, Kute? All the heritable factors you have in doubles, right? Diploid, no, right? So you have doubles. But when you make the gametes, they separate and go into the gametes separately. That is called as law of segregation. Segregate means you separate and go, right? A and A doesn't go as a packet to one gamete. They separate and enter. That is the first law, right? So he work out how it happens. You take sperms here and eggs here, right? Now this is the purple color plant, right? And it has two genes here, and here you have the white color plant, right? Now you take the purple color plant, it has two genes. What are those two genes? Capital A and capital A. So all the sperms that are produced by the purple plant carries only capital A. All the eggs that are produced by the purple plant again carries only capital A. So all the sperms and eggs in the purple color plant carries only capital A and capital A. All the sperms, all the eggs carries only one capital A allele. You take the white color one, it has the simple A and simple A. So they segregate at the gamete formation. Gamete හදද්දි මේවා segregate වෙලා යනවා, right? At the gamete formation, these alleles separate and eggs get the simple A, sperms get again the simple A. All the eggs and the sperms produced by the white color flower plants, they have only the A and A. Simple A and simple A. Right? Now in crossing, monohybrid cross, what happens? A sperm from the purple crosses with the egg of a white color. So that progeny, that plant is having capital A and simple A. Again, an egg of a purple flower can fertilize with the sperm of a white color one. So the zygote is capital A and simple A. Right? And the purple color one, it produces, here in this case, purple get capital A, simple A. Right? And in this case, again, capital A and simple A. What do you mean? All 100% of the progeny is having only heterozygous capital A and simple A. That is why if one generation is producing only purple color plants, right? So you don't want to draw this sort of square to decide that, but I came to that level also to understand how the F1 generation became as the capital A and simple A, right? Now, the second step of the experiment is to cross purple ones and this one. So there, all the purple color plants are having capital A and simple A. Now the thing is different here. Now what happens, okay? Again, we'll draw this square. Can be either sperm or the eggs, right? Here you have the purple, here you have the white. So again the purple, right? All the plants are purple, no, in F1, right? So here the purple color one is having a genotype of capital A and simple A. This one is also having capital A and simple A. So the sperms in this plant can carry A. Some of the sperms carry capital A because they are segregated, and some of the sperms are carrying simple A, right? Some of the eggs are carrying capital A in this plant, and some of the eggs carry simple A in this plant, right? So either way, the egg is fertilized with the sperm, right? So one combination that can happen is capital A binds with another capital A, sperm and egg. So a plant can originate containing capital A and capital A. So in this fertilization, both the sperm and the egg carries capital A. And in other fertilization, simple A can be crossed with the capital A of this plant. So you get a plant having A from a sperm and A from a egg, right? In this case, simple A from a egg and a capital A from a sperm, again you produce capital A and simple A zygote. And there can be an instance in one plant, sperm is also carrying simple A, egg is also carrying simple A. Understood? Right. These are the four possible genotypes that you can get in this monohybrid cross. When you cross a heterozygous F1 plant with themselves, right? These are the possible four outcomes in genotype, right? So, what are the genotypes? Are there four types? Even you have four boxes. What are the genotypes that you have here? You have capital A, you have capital A and simple A, and you have simple A and simple A. So, three types of genotypes are there. Capital A and capital A, there is a one chance of having this one. You have only one here. Right? Capital A and simple A, having capital A and simple A in a zygote, there are two chances. Because either way that can happen. One plant is having a capital A sperm, simple A egg. The other chance is capital A egg and a simple A sperm. So there is a two chances of having this combination, capital A and simple. And having the simple A in both the sperm and the egg, again you have one chance. Right. Now look at this F2 generation genotype. You can have homozygous dominant alleles, capital A and capital A, one chance. Heterozygous state, capital A, simple A, two chances are there. Homozygous recessive alleles, you have one chance. Simple A and simple A. This is the genotypic result that you can have. This is how Mendel explained this. So if this happened really in the cells, what could be the phenotype? Now, what is the phenotype of capital A and capital A? It is purple color. Capital A and simple A, again, the phenotype, purple color. So, both these are purple. And this one, simple A and simple A, is white. Right? So, how many chances of having a purple color plant in the progeny? 1 plus 2 is 3 and 1. Right? So, in genotype of a monohybrid cross in F1, in F2 you get 1 to 2 to 1 proportion of genotypic presentation. And if you take the phenotype, you get 3 to 1 proportion in the progeny. Right? This is how he got this result in his experiment. In the F2 generation, he got 705, but you don't want to remember these values, 705, 2 to 4, you don't want to remember, just get an idea, but you have to remember 3 to 1. So his result was 3 from purple or the dominant allele and 1 from the recessive allele trait. Dominant trait 3 times, recessive trait 1 time. How it happens? In this way. So he explained his observations in the experiment by explaining the scenario in a paper. Right? So in a paper, he drew this square and this is called as the Punnett square. Punnett square. Right? And he said, so in the Punnett square, what is the basic thing that he explained? All the heritable factors separate or segregate and goes into the gametes at the gamete formation separately. This is the first law of inheritance by the Mendel. So you call it first law of inheritance or the law of segregation. Law of segregation. Understood? Right? This is simple as that. So this is the first law, Gregor Mendel's first law. Right? To explain that thing, he used the Punnett square. Okay, right. Easy, no? Easy, no? Right. Shall you write this down? So you learn the first law of the Gregor Mendel. So first law of Gregor Mendel, that is the law of inheritance or the law of segregation, is explained by a monohybrid cross. Mekedi, he used only focusing one character, color of the flower. Ek character kai focus kare. Right? He focused only one character. Likewise, he did lot of experiments focusing only one character. For example, this is focusing the color of the flower, and he did focus in the shape of the seed, and he did some experiment focus in the length of the stem. Right? Some are having long, tall stem. Some are having dwarf, short stems. So he did those. Right? And what else? He did some things, the position of the flower. Some flowers are originating at the end of a stem, axial ones, or some are having the terminal ones, or the some are coming from the buds in the middle of the stem. So axial flowers and the terminal flowers, right? Likewise, he got about six characters he focused and did lot of experiments. In all these experiments, where he focused only one character at a time, the result he got was 3-1 in the F2 generation. So that is how he came to the conclusion of law of segregation. Each and every time when the gametes are formed, heritable factors segregate and separate and goes to the gametes, either the eggs or the sperms. So one plant, assume that this purple plant, you take one plant, you produce 100 sperms, 50 is having capital A, 50 is having simple A. Same plant produces 100 eggs, 50 is having capital A, 50 is having simple A. That is how the F2 generation comes as 3-1 proportion according to this explanation. Right? Okay. Understood? Any questions, Kute? No questions. That means clearly understood. Okay. Shall we go to the tute? While we are completing the tute, if you get a question, you can ask me. Huh? Hmm. Right. Monohybrid cross. Now we are going to elaborate on monohybrid cross, Kute. Right? Right. Mendel derived his first law of inheritance. First law of inheritance. First law of inheritance by following only a single character in one breeding experiment. By following only a single character in one breeding experiment, such as flower color. He started by crossing pure breeding parents. Pure breeding parents. Remember, every time Mendel start the experiment with pure breeding parents. I don't know who told that without the knowledge of anything about the genes or the chromosomes. Just we are starting crossing the purple color plants and white color plants. We don't have that knowledge of getting a pure breeding ones because we don't know about a gene, we don't know about inheritable factor, but he started with that thing. Sometimes a God has come and told him, "මෙහෙම කරපන් කියලා." Right? So the Mendel derived his first law of inheritance by following only a single character in one breeding experiment, such as flower color, and he started by crossing pure breeding parents with contrasting traits. Pure breeding parents with contrasting traits. Contrasting traits. Contrasting traits. Mendel presented his all these experiments and findings and these laws in about 1866. 1866, that's 1866, he presented this thing. And at that time, he was not appreciated. He was not admired with these presentations. But later on, about 1890, after about 20 years back, right, the gene and the chromosomes were found. And then only the scientists understood that the Gregor Mendel what he told 20 years back was correct. And then only he was appreciated and given all this uh, appraisal, right, after 20 years of his experiment. And then only he was considered as after 20 years, considered as the father of genetics, right? So what is the lesson that you can have it here? Now, if you find anything new to the world, right? Now, if you are sure about that thing, the world may not admire it, world may not appreciate it at the time, but you still, if you are sure about what you have given to the world, to the society, you stay in that position and keep going. One day, the people will understand the truth and they will appreciate it. Right? Sometimes you may not be alive at that time. Still okay. All the credit will goes to you even after the death. Right? So don't be hurried. Do something to the society. I mean, I'm do something, right? What I want from you is not to become a common medical officer, common doctor, common scientist, right? All the doctors there are a number of doctors are there, but very good quality. The society changing, right? The system changing doctors are very few. So, what I expect from you is to become a character among the doctors. Okay, right. So, next point. All the F1 progeny, all the F1 progeny produced, all the F1 progeny produced from pure breeding parents are what? Monohybrids. They are monohybrids. Monohybrids. What are monohybrids today? They are heterozygous for one gene. Monohybrids. So all are capital A and simple A. Meaning that they are heterozygous. Meaning that they are heterozygous. Meaning that they are heterozygous for the particular character being followed in the cross. F1 hybrid pea plants were then self or cross-pollinated, and F2 generation was produced to explore the traits resulting from a monohybrid cross. That is a monohybrid cross. Monohybrid cross. Right. So I have given you the experiment that he did in a diagram, in a schematic form, and that diagram is given in your note also. Right. So purple flowers crossed with white flowers, and these are pure line ones, and therefore all the F1 generation produced was 100% purple, right? And then these monohybrids he did a self or cross pollination that is called as a monohybrid cross, and then this is the observation in the F2 generation, right? F2 generation, 705 purple flowered plants were there, and 224 white flowered plants were there, right? That means the proportion is 3:1. Now he wanted to explain this, how this happened, right? So Mendel, during his experiment, crossed pure breeding purple flower plants and white flowered plants. Then he allowed the resulting F1 hybrids to self and cross-pollinate with other F1 hybrids. Finally, he observed the F2 generation plants for the color of flowers. During his observations, Pute, during his observation, all F1 plants produced, purple color flowers, all F1 plants produced purple color flowers. However, in the F2 generation, both purple and white flowered plants appeared in a ratio of approximately 3 to 1. In a ratio of 3 to 1. Remember this 3 to 1. Right? So now today I am telling again, right? Keep this in golden rule. If it is a monohybrid cross, right? If it is a monohybrid cross, P generation is contrast PO lines. F1 generation is 100% of the dominant trait. F2 generation is 3 to 1. Right? Now, in the exam, questions come from the bottom line. Now, we'll do some questions, right, after the theory part. The question starts saying that in a breeding experiment, the F2 generation found to have tall stems to short stems in 3 to 1 proportion. Now, when you see this 3 to 1 proportion in F2 generation, that means you should remember that, you should realize that this is a monohybrid cross. 3 to 1 phenotype එක කිව්ව ගමන් මතක් වෙන්න ඕනේ මේක first law එකේ question එකක්. This is a question in the first law and it is a monohybrid cross. Right? Likewise, if they are given the genotype of the F2, of the F2 is 1 to 2 to 1, that means again monohybrid cross. First law. Right? That is how you work out on questions. Right? Now, in the tute. Among the heterozygote resulted in the F1 generation, the heritable factor, Mendel used the heritable factor, not the gene, right? The heritable factor responsible for producing white flowers were suppressed in the presence of heritable factor that produced the purple flowers, right? As a result, the heterozygotes were all producing purple color flowers. That is how the F1 generation is producing all the purple color flowers. Therefore, the heritable factor for purple color, flower color, is dominant. Therefore, heritable factor for purple flower color is dominant to the white. Dominant to the white. So, heritable factor is dominant allele and the purple color is the dominant trait. So, within bracket, dominant trait. Phenotype is dominant trait. Accordingly, the factor for the white flower color is referred to as the recessive trait. White color is referred to as recessive trait. Recessive trait. Mendel observed that the same pattern of inheritance consistently occurred in 6 other characters. So not only the color of the flower, he saw that the same type of distribution happens in contrasting other characters also. What are those characters? Position of the flower, color of the seed, shape of the seed, right? shape of the pod, right? Color of the pod. Pod means that the covering of the seeds. Mungata karalakian, that is the pod, right? So shape of the pod and the color of the pod and the length of the stem, short or these are the characters that he used for contrasting traits, right? Okay. Now, how we presented Mendel's first law of inheritance, that is called as law of segregation. The both names are referred to the same law. Either you can say first law of inheritance, Mendel's first law of inheritance, one name. Another name given for the same law, Mendel's law of segregation. Law of segregation, this is referring to the first law, right? Okay. Right. Mendel's first law was put forward to explain 3 to 1 inheritance pattern observed among the F2 of Spain in his monohybrid experiments using the pea plants. As per his hypothesis, right? එයාගේ hypothesis එකක් තිබුණා. This one, right? What is his hypothesis? We'll write. Each heritable factor each heritable factor each heritable factor is determined by each heritable factor is determined by sorry should be I made a mistake. Each heritable character, sorry, it should be the sentence should start with each heritable character. Character කියන්නේ color of the flower කියන එක. Each heritable character is determined by two heritable factors. Two heritable factors. Each heritable character is determined by two heritable factors which are known as alleles. Which are known as alleles. Okay. So that is a hypothesis of it. The hypothesis means you assume that this may be the reason. Now, these are scientific hypotheses, are very important ones, right? So these are the ones who has changed the whole world, scientific world, the destiny, right? As per the hypothesis, what is the hypothesis of the Mendel's? Each heritable character is determined by two heritable factors, which are known as alleles, right? So during the formation of gametes, make it my important place, right? During the formation of gametes, the alleles for heritable characters are separated. In the formation of gametes, these are separated. During the formation of gametes, the alleles for the heritable character are separated. Separated. All these alleles separated and get into each of the gametes formed. So they are separated and get into each of the gametes that you formed. Each of the gametes means each of the sperms, each of the eggs. Right? This is now known as Mendel's law of segregation. Mendel's law of segregation. Or Mendel's first law in inheritance. Mendel's first law in inheritance. Inheritance. Right. Okay. So that is the first law. Right. Analyzing genotype and the phenotype ratios using a Punnett square. Right. How you can explain how this phenotype came. So you explain using a Punnett square. This is a Punnett square, right? In pea plants, Pute, Mendel observed two different traits based on stem length, tall and dwarf. So now you are taking another example, tall and dwarf, right? Now take when you examine the length of the pea plants, the stem, some are having long stems, some are having short stems. So two contrasting features. Now you are going to explain these 3-to-1 phenotype by using these tall stems and dwarf stems in this example, right? In pea plants, Mendel observed two different traits based on stem length, tall and dwarf. For his experiments, pure breeding tall, shall I draw this one? Otherwise, you will get confused. No? I took the color of the flower here, so I'll take that thing also. Tall and dwarf. For the better clarification, I'll take the same sample. You take the tall stem and cross with dwarf plants, right? Tall stem and the dwarf stem. So this is the T generation. And what happens in the F1 generation? All the progeny was tall, right? 100% tall. So this is the F1 generation. So what he did? Tall F1 cross with another F1, monohybrid cross. And you got tall plants as well as dwarf plants into 3-to-1 proportion. 3-to-1 proportion. Right? Tall plant, pure breeding tall plant, pure breeding dwarf plant crossed as the P generation. Then all the F1 generation was 100% tall plants. Then this is a monohybrid, right? So monohybrid cross was done. Tall plants in F1 self or cross pollinated and got the F2 generation. In the F2 generation, right, tall to dwarf plant proportion was 3-to-1, right? So what has happened? Now, F1 generation 100% tall, that means tall is dominant. So dominant trait is tall. So dominant gene is capital T. So this is pure line. So this is capital T and capital T. Dwarf means simple T, simple T. So 100% of the F1 generation is heterozygous, capital T and simple T monohybrid. So this is a monohybrid cross means capital T, simple T crosses with capital T and simple T. And you get this 3-to-1 phenotype. How you get the 3-to-1 phenotype is by this way, right? Capital T, simple T, in the Punnett square, capital T, simple T. So here you get capital T, capital T, capital T, simple T, capital T, simple T, simple T, simple T. This is how you get the genotype proportion as 1 to 2 to 1. Okay, right. You have asked questions. Sorry. So I was wondering, you can't see, I can't see you, sir. Sorry. What has happened? I am muted? Oh, no, that is the previous one. Gag, you can see now. There are some blurring happens with the camera, no? Why is that? No, everybody can see now. Have you lost some part? There is some lighting issue here. Maybe that thing. Now it's clear, no? Okay. Right. Sorry. Sir. Now the blurring. Not the blurring you had. Screen share. Sorry. Yes. Right. Sorry. And I put that doom. Take it. I forgot to remove the screen share. Now it is clear, no? Parent plant, PO breeding, tall and dwarf, and you get the F1 generation 100% tall and monohybrid cross happens, 3-to-1 phenotype presentation. How you get the 3-to-1 phenotype presentation is explained by using a Punnett square, and this is how. These are the possible genotypes that you can get. Capital T is one type. Capital T, simple T is another type. Simple T, another type. So in monohybrid cross, genotype proportion is 1 to 2 to 1. Remember that. Phenotype proportion is 3 to 1. Right? Remember it as it is written in a stone. And remember. Okay. Shall we read the note again? This part, huh? I'll share the screen. Analyzing genotype and phenotype using phenotype ratios using the Punnett square. So Punnett square, why we use Punnett square? Just to explain how the 3-to-1 comes. Right? So in pea plants, Mendel observed two different traits based on the stem lengths, tall and dwarf. For his experiments, pure breeding tall and dwarf. Pure breeding tall and dwarf pea plants were selected for cross-pollination. For his experiments, pure breeding tall and dwarf pea plants were selected for cross-pollination. Therefore, F1 generation was self-pollinated. Therefore, F1 generation was self-pollinated in order to obtain F2 generation. Therefore, F1 generation was self-pollinated in order to obtain F2 generation. Right. During self-pollination of F1 hybrids, gametes carrying different alleles fuse randomly. right? They fuse randomly. Such random fusion of gametes produces zygotes with four genetic combinations. Four genetic combinations. Fuses with four genetic combinations. Right? Okay, look at this one. These are the four genetic combinations that can happen when the gametes, that means eggs and the sperms, are fusing randomly. These are the possible ways that can happen, that fertilization can happen. So, possible four genetic combinations can happen. Okay, right. Four genetic combinations. A Punnett square, a Punnett square P-U-N-N-E-T-T. Double N-E double T. A Punnett square can be used, Punnett square can be used to illustrate these genetic combinations. A Punnett square is a graphical representation. Punnett square is a graphical representation. It's a graphical representation of the possible genotypes. Graphical representation of the possible genotypes of an offspring. Of an offspring. So Punnett square is a graphical representation of the possible genotypes of an offspring arising from a particular cross or a breeding event. In one breeding event, these are the possible outcomes that can happen. So the graphical interpretation is done by this sort of square, and that is called as the Punnett square. Right? An example is given in the following figure. Right? So I have given you the figure there. Okay. Now you learn the first uh, theory, first law of inheritance, that is the Mendel's law of segregation. Right. Any questions up to now? No, we'll stop from there. Right. So in Friday, we'll start from the dihybrid cross. That means the second day we'll learn the second law of inheritance, and then some of the diseases which are related to these ones. Now, not like in the other cases, in genetics, diseases are also very, very important because a lot of questions asked from the diseases also, right? So these diseases, genetic Down syndrome and so on is Klinefelter syndrome. So what is the genetic abnormality there? You have to understand and remember. So you can answer the questions there, right? Up to this place, right? Now, today you learn the very basics, started from the very basics of the genetics, right? You learned about the Gregor Mendel, and you learned about how we plan the experiments. And then we learned the vocabulary which is used throughout the genetics, in not only in the advanced level syllabus, but when you go to the medical faculty or the science faculty, still the genetics, you have to know this basic vocabulary. Now you are armed with those technical terms, right? So you learn about the technical terms or the vocabulary, and then we started learning how the monohybrid cross was done by the Mendel and what are the observations that he got. He got pure lines in the P generation, and F1 generation was 100% the dominant trait, and F2 generation dominant trait and the recessive trait was there in a 3-to-1 proportion phenotype. Dominant trait 3 times, recessive trait 1 time. 3-to-1 proportion in the phenotype. That was explained by using a Punnett square, right? Using a Punnett square. And there, the possible genotype outcomes, there are 4 possible combinations are there. 3 possible genotype outcomes are there. homozygous dominant, heterozygous, and homozygous recessive. That proportion is 1 to 2 to 1. Those are the things that you have to remember. Any questions from that part today? Right. If we don't have any questions, we'll proceed with the next day, Friday, right? So every Monday evening and Friday evening, we'll do the classes, right? And I have arranged the 8 classes for this month, no, 6 classes. And by the time, suddenly, if I got a free time, I'll put some additional classes also, other than these 6 classes, right? June, anyway, we are going to have 8 classes, right? More than 8. July, again, more than 8, and we'll finish up this thing. Don't worry, genetics is the most important part there. Then the DNA recombinant technology and the environment technology, the environment biology, those are not, a lot of questions are not coming from those units, right? So don't worry about that thing. And by the time we'll start doing some, after finishing the genetics, we'll do some full paper classes. So you will cover the whole syllabus. Okay, right? Any issues? Okay, then we'll stop it and have a nice day and a nice week. Hope you will study thoroughly. මේ දවස්වල හොඳට පාඩම් කරන්න, right? හොඳට පාඩම් කරන්න, time table එකට පාඩම් කරන්න සහ and one important thing, හොඳට නිදා ගන්න, right? You have to have a good sleep, otherwise you will not remember most of these things. So at least 6 to 7 hours is needed. Preferably 8 hours is very good. right? අනිත් ඒවට යන ටයිම් අඩු කරන්න. ඔය ෆෝන් බලන ඒවට, ටීවී බලන ඒවට, එහෙම ඇවිදින ඒවට ක්ලාසස් පුළුවන් තරම් ඔන්ලයින් ඒවට කියනවා. යන්න එපා කියන නෙමෙයි ෆිසිකල් බට් අර ට්‍රාන්ස්පෝර්ට් ටයිම් traveling time එහෙම අඩු කරගන්න අපරාධ එක wastage of time at this stage, right? So you productively use current time එක and have a good sleep, good rest, and then study. And have a good meal also. හොඳට කන්න උත්සාහ. Water intake again is very important. හොඳට වතුර බොන්න ඕනේ. හොඳට nutritious diet එකක් ගන්න. And have a productive 2 months or 3 months in these 3 months time. Okay, right. See you then. Bye.